Okay, so this was the activity that we were doing yesterday, and I just want to explain it again further. So on the bottom here, you're supposed to represent homologous pairs. So I said pink and red were homologous, and then green and yellow. So during meiosis, the first part, homologous chromosomes will line up together. So each being is representing a chromatid, and the whole thing together is representing a chromosome. So I have my two homologous pairs. So the first part will be that the homologous pairs will separate from each other. So now this part will represent one cell, and then this will represent another. So that's how I determine these. So then this will go into that cell, and etc. for the other. So now the second part, so this was really meiosis 1. Meiosis 2 is when chromosomes split apart into chromatids. So the chromosomes don't line up with each other because, again, they're not homologous if you look at my chart. So my chromosomes will line up together and they will then split in half. And now we have our chromatids during anaphase. And so then those will make our new cells. That is the activity from yesterday. Now I want to go over one other thing, independent assortment. So here I have my homologous chromosomes lined up, and that's what the result was. Now what I can do is the alignment could change. And notice what happens. My pink and green separate into one cell because, again, homologous separate. My yellow and red separate into another. So now the result of my new cells will be pink and green and yellow and red. So they will line up during metaphase. The chromatids will separate during anaphase. Cytokinesis will occur, and those cells will develop into our gametes. And then the same thing will happen to our cell below. So the chromosomes will line up during metaphase. During anaphase, they will split apart. Each of these will become a new cell. Notice my genetic combination different from the first one. That is called independent assortment.